So the next speaker, Christopher Hart. Chris is the co-founder and CEO of Creyon Bio. He led Ionis Pharmaceuticals genomics department, pioneering company-wide initiatives in rare and common diseases. With a PhD from Caltech and postdoc training at Yale, he blends research and teaching in bioinformatics. As a visionary leader, he advised the White House on diverse topics before shaping the future of biology and technology at Creyon. So,
thanks everyone for sticking around, and thanks for the kind introduction and opportunity to talk. So I think I'm going to, where I'm going to come in is I'm going to start talking about what we're building at Creyon. I think it connects very nicely from all the stories we've heard today. And as we work forward, I think one of the things that probably highlights why we're all here today is really this notion of what modern biology really gives to us. And I think Francis Collins nailed it about a decade after he sequenced the genome with the public genome savings project, where he says perhaps the most profound consequence of the genome revolution in the long run will be the development of targeted therapeutics based on a detailed molecular understanding of pathogenesis. This is actually why we're all here really today. It's because we can, in fact, understand at a foundational level what's driving pathogenesis, both for common diseases often, but also for rare diseases. And I have a very similar slide to Winston's slide where we're looking at this long tail of rare diseases. And I think this is actually the product of exactly what we see with modern biology. So we really can understand the genetic basis of disease, and it drives to us understanding that there's a reason for the rare disease crisis, and maybe there's hope that we could actually start to address some of these underlying pathologies. Francis also highlighted really what's holding it back, though, which is it's not our lack of desire or hope or want to sort of build therapies in this place. It's really that it's hard. And he coined it here very, very eloquently, where he says this is also the goal most challenged by long-time lines, high failure rates and exorbitant costs. So when we think about how do we start to make inroads to developing therapies across this long tail of rare diseases, obviously we're thrilled to be part of an ecosystem here in San Diego and more generally globally, where people want to collaborate, where there is a lot of interactions to go forward. But I think we need to also think very deeply about what are the foundational challenges that are blocking us from making strong process here. And I think there's an elephant in the room, and it's the same elephant in the room for common diseases as well, which is that it takes too long and costs too much to make too few drugs. And if we extrapolate that out, and I think it was said by multiple people here today, if we just extrapolate out the timelines it takes to make a drug, and we look across that long tail, well, none of us want to face a millennia while we wait, and nobody wants to spend the GDP of the planet to make these drugs. So we really have to think, how do we foundationally lower those costs? How do we make it more efficient? And how do we make it so that we don't have to raise millions of dollars to build these drugs? And we've heard many talks about the promises of oligos, so I won't dwell on it, but the real value of oligos is that they do have this capacity to intervene at the foundational processes that drive molecular biology. We know that we can engage directly to modify the expression of genes, either at the pre-mRNA level, the mRNA level. We can change how they splice. We can edit genomes. We can introduce new things. We can even change how the ribosome engages. We can change how proteins interact with molecules. What's the commonality here, though? We heard this notion of a programmable medicine. I think that's a fantastic term. What makes them programmable, though, is that they're actually just polymers. They're polymeric molecules. They're not small molecules. And as polymeric molecules, there's a thing that makes them really interesting as drugs, which is that the base sequence that we use to interact with the targets of interest, most often that happens to be a nucleic acid like an RNA, that's actually separable from the chemical modifications that we put on these molecules to make them into good drugs. That separability is actually what makes them programmable. That's what makes them possible to be put forward. And the thesis that Creyon's put forward is that you can think about it as programming, but I think it's more generally, and these are hardware. This is an engineering problem. How do we actually solve this as an engineering problem? How do we make these polymers engineerable? And when we think about that, then we have to think about what is the thing that we need to engineer most what is a sort of an invariant property of drugs that we always want to make better? I don't think anyone's going to ask you to make a drug less safe, with the exception of maybe some chemotherapeutics, but they want those safe as well. They just want them targeted. And that's what's really been, I think, limiting the field, is that we've seen amazing success stories, and I think we all can stand on the shoulders of our history and the past and say that there is huge potential with this modality, but we can't look at it and not say that there's an issue, which is not only when you change the base sequence, it may be toxic. It may not be active, but it may be toxic, and you don't see that until you actually put it in animals, because those toxicities emerge actually as organ level failures. And this is something that's plagued the field and really held back our capacity to push these drugs forward. So how do we get to a place where we can solve that problem? And when we started Creyon, it was really about seeing this as the opportunity, which was we should be able to engineer these compounds, but we don't know how to engineer them, and we don't know how to engineer them because we haven't collected a sufficient amount of data or the right data to actually address the things that actually hold back the field. And I'll sort of give you an idea of why it's been so hard to collect this data. And it's because the space is ridiculously large. And for those who have thought about designing oligos before, you've probably already stumbled upon these numbers. For those who have just started to think about this process, most oligos are somewhere between 16 and some odd bases long, but they're short. But they're composed of 4 bases, A, C, T's, and G's. Even if you go in the shorter end, 16, that's 4 billion possible combinations. That's a big number. But then we can't just put a DNA and RNA molecule into the cell. The cell doesn't like it, has a lot of adaptations to both get rid of it, or worse, we'll think it's a foreign DNA and have an immune response to it. So we chemically modify them. And we chemically modify them in different places. And I show here a molecular structure. For the scientists among us, we've all thought about it before. For those who are new in the space, we have bases over here. These are the things that allow us to make Watson-Crick-Franklin hybridizations to the target genes of interest. We typically modify the sugar that the base hangs off of, and we typically modify the backbone. And sometimes we even modify the base. Every time we make these modifications, the key thing that we need to recognize is that they're actually not very far away from each other. And if you were an electron hanging out in this sugar, you're going to be impacted by whatever this base is. And likewise, if you're an electron hanging out in this backbone, whatever the sugar is going to change properties. And what we know is that the electrostatic properties, or where electrons like to be on these molecules, impact what proteins they like to bind to, what proteins they don't bind to, how they aggregate. And those interactions are the things that drive the pharmacology, whether it's active, whether it's safe, whether it migrates to the cell. Those are not independent now. Now we have this collusion, this deep collusion between the sequences you use, which we know we have to modify because that's how you're going to target your gene, with the chemistries you use to improve the pharmacology. The field's been hoping that maybe there's one sugar or one backbone that'll make it safe across all sequences. We've yet to see that to be the case. In fact, it hasn't been the case. It probably never will be the case because they are, in fact, entangled. They're stuck together. And just to follow up on my combinatorial problem, that means that you have to understand how when you make these modifications, they're going to interact together. And that means that you're dealing with, like, even with simple things, 10 to the 24 possible molecules to construct. Now, if you're thinking that doesn't seem so big, it's huge. It's like the number of stars in the galaxy. It's a big number, which means that if you're thinking about what can I use, what data can I use to learn my way around this space, you're going to be screening for the rest of time before you even come close to approaching a place where you have enough information to actually extrapolate. So we have to think deeply about how we find this. And that was one of the first things we recognized at Creyon, is maybe there's a way to survey this space in a way that was a little bit more efficient, but it required us to think a little differently. And I think this is one of the critical things that we've thought about, which is a lot of times, I've heard it multiple times today, there's no business model in this space. The long tail means that, these patient populations are too small. And it's because people haven't thought about how do we actually integrate across that tail, such that we can combine that revenue. And that's where you get a business model. But the way you do that is that means you have to be able to actually distribute your costs across that long tail of diseases and distribute your workload across there. Sometimes you'd hope that the same drug might work for multiple diseases or work for a common disease. But as we've heard, sometimes you need a little selective drug. Sometimes these modifications or these variants are exceptionally rare. Maybe one, maybe a few. that means the same drug is not going to work. It's not going to extrapolate. So you have to think about how do you integrate these efforts across multiple programs so you can then share that workload, so you can distribute the cost but still integrate the revenue. And that's really where we are. Creyon is we're thinking about how we do that. And we do it by changing our perspective. We do it by thinking about how do we solve this engineering problem? How do we change making oligos from a discovery effort to one in which you're actually engineering compounds that have favorable pharmacology? And just to sort of highlight the point and why it's so important, and this is something that I think is critical for anyone walking into the space to understand, which is that the sequencing chemistries do in fact collude quite a bit. I have a little data slide here. It's 96 oligos targeting a liver-expressed gene in mice. None of the details all really matter, except that they're all made with the exact same chemical motif. And for those in the space, this isn't all that surprising, but what should be highlighted all the time, which isn't always recognized, is that these compounds along the x-axis is knockdown in vivo in livers, and then on the y-axis is a measure of ALT, which is toxicity. If you go above 100, it's considered to be pretty bad for the mice. Once you get up to, and that'd be a log 10 of 2. And you can see very, very clearly what comes out is that there are compounds, same chemistry again, just different sequences that look terrible. I'm going to tell you this isn't because they're making some off-target binding to some other gene and knocking down some like essential gene. It's just because they're binding to something in the cell that makes the cell unhappy. I don't know what it is. I just know it makes the cells really bad. I want to avoid those. And then I can tell you there are other genes, other oligos, same chemistry, different sequence that look like saline, except they knock down this gene. I want more of those. I don't know why they're so safe. I don't know how they get in there. But what I want to understand is what are the chemical properties that have been imparted into those molecules when I put new chemistries on with some aspect of the sequence that produce this biophysical property that allows it to sneak into the cell, do what it's supposed to do and nothing else. So that's what we need to figure out how to do. But the key thing here is that sequencing chemistry collude. And they collude here when we're sort of pulling chemistry constant, but they also collude if we do the opposite, which is say I have a sequence and I just put on different chemistries. Sure enough, we can have the exact same problem. So in this particular case, I took a drug that was targeting a gene, and I put it into a mouse. This is a 3-10-3 lock nucleic acid. For those who are in the space for a while, these were historically thought to be pretty toxic in the liver due to some publications that came out at some point. It turns out there's just a collusion that was not appreciated. They're not all toxic. It's just some of them are toxic. But when you put them in the brain, you can have things that look actually pretty clear. But one of the things we had to think about is how do we assess toxicity in the brain and be very careful about assessing for toxicity in the brain, because last thing you want is have neurons dying, and you have to be very sensitive about how you see those. So when we do that, we actually look at tunnel staining, where we can look at apoptosis in the brain at a very high dose, and we see these compounds are exceptionally well tolerated. Generally, if you choose them well and you design them well, you engineer them to have properties that make them well tolerated. But if you take that same sequence and make it a 5-10-5 mode, which is, there's lots of safe 5-10-5 modes out there, but they're not all safe. In fact, this one actually causes quite a bit of neuronal death at half the dose of the LNA compound. That's concerning if you're not paying attention to the fact that the sequence and the chemistry collude. You can't just assume you're going to change the zip code and magic's going to happen. These are foundationally different molecules every time you change either a base sequence or a chemistry. And you have to understand what you're doing by figuring out those design rules, those engineering principles. And that's really what we've been building. The sort of the foundational idea here, though, is how do we think about taking this notion of a polymeric molecule and turn it into truly an engineering platform. So we've been spending a lot of time creating these really bespoke data sets where we can learn. These data sets are not meant to teach us to make a drug. These data sets are really meant to inform models that then allow us to connect the biophysical properties, really from a quantum first principle, to the pharmacology that we observe by putting these molecules into mice and observing with very detailed assays. And these are like IND-like studies, but at the end of the day, what we got to is this place where our models allow us to improve the random hit rate of getting a safe oligo by at least 30x. That has immediate benefits in how fast you can go from I have a target of interest, a allele I want to target, to a drug that I might be able to put into a patient. And I'll show you a story where we were able to make a drug using the platform in about 13 months. We've done that more than once. We also use that same platform. This is where I think we get to, how do you think about the business model? Well, this same platform works for making common disease drugs as well, because we want to modify common diseases just like we do for rare diseases, they're just different genes. We want to be able to program modifications of genes that have pathology, and we can do that with the same sort of basis if we understand how to engineer these compounds. And there we've handed over some compounds for a neurodegenerative disease for partner, and then we have new compounds coming up for a common disease, and we've repeated this process. It takes about 4 months to go from a start to a couple of leads. The other aspect, which I'm not going to talk today, is about these compounds are amazing. They can get into cells and do great things to modulate gene expression, but they only can do that if they get into the cells. So we've also been working on how do we make access to different tissues and cells also programmable. And our core competency is really about how do we engineer polymeric nucleic acids. So we made aptamers. Aptamers are just slightly longer aptameric extensions. We sometimes think about them. These are exactly folded sections of an oligo that you put at the end, and they fold up into things that can have high binding affinity to receptors on cells in a very specific way, so they can get pulled in to the right cells at the right time. Ideally, what we're aiming to do is really unlock both common and rare diseases by being able to sort of engineer these compounds in a way that is differential from the way that you might think about discovery. The sort of one layer deeper into terms of how the platform works, I sort of alluded to this, is that we spent a lot of time, and this is another thing that I was actually a little surprised, but when we started thinking deeply about what drives the properties of these molecules, we realized it was in fact the electrostatic properties of these compounds. Electrostatic properties are where electrons live, and then you go and you talk to your computational chemist, and like, that's cool, that's a quantum problem. You got to understand the quantum nature of these molecules, but then you realize the challenge there, of course, is that you can't really simulate a whole polymer at that sort of level. So we had to spend a fair amount of time building up ways of stitching these molecules together. So we could take a quantum resolved model and stitch it together into a larger molecule that would still project out biophysical properties that were meaningful in terms of the pharmacology of these types of medicines. And that's an interesting academic thing we did, but it was only the point in which we were able to actually connect it then to hardware and pharmacology data sets that we created with a very purposeful intent to inform models. So here we did thousands of IND-like rodent studies where injected compounds that were pretty, they're pretty useless by themselves, but in aggregate, they built this platform that allows us to engineer these compounds. One of our investors, when I first showed him the set of data that we created, we'd done our first set of, I think, a couple hundred compounds, put them into mice, did this ridiculous experiment, really bet the company on it, showed it to our investor, and he came down, he was like, "Oh, that's great. Chris, you just did the largest mouse study I've ever seen, and you still don't have a drug." And I was like, "Yep, that is true. I don't, but I have something better. I have a platform, and I can engineer drugs. I'm biasing ourselves to making drugs, and part of their mission is to invest in the Star Trek future rather than the Star Wars future." So we were on a path to making a radical abundance of gene-centered medicines, in the words of Matt Aka, the founder of TCBC. So we put that forward, and we couple, of course, this huge amount of data from rodent studies with, rodents are great to understand mammalian toxicities, but we do worry about human things. So we also put these oligos into in vitro systems where you can look at things in tissue culture, so we can understand human cell culture experience. But then the real crux that you have to worry about a little bit sometimes is that immune systems are different across all species. So we also spend a lot of time doing ex vivo experiments with PBMC assays to understand what the immune impact of these compounds would. And you can't do that in a monkey, you can't do that in a dog or pig or mouse. You have to find something that's humanized. So that's why we have these ex vivo experiments. Plus, when you're in vitro, you can do dose response and time courses that you can't afford or think about doing in animal studies. But this data together is what ends up aggregating, and then we use very, very purpose-built machine learning tools to solve what I think is really a problem that's perfect built for machine learning, which is it's an engineering problem, which is how do I connect a feature vector of biophysical properties on these molecules to pharmacology that I observe in vivo, so I can then understand what properties I want to impart on my molecules to have the favorable pharmacology that we want on our drugs. And by doing that, we now can say, for any given compound, here's the sequence I need. What would be the right chemistries to impart a safe drug? And with that, we could start to make drugs. So I'll tell you one story of making sort of a allele selective drug for a rare disease. It's a story that's too familiar. but is also a story of hope, I think, which is this family was able to get diagnosed about 1 month of age. They enrolled in a rapid whole genome sequencing study. They got sequenced and they were identified to have this single nucleotide variant, exceptionally rare. There's one other individual in the world that had been identified with this particular variant, and it was a nuclear transporter. And that nuclear transporter kept things in or out of the nucleus, and it was particularly interesting because it was effectively causing things to be willy-nilly moving in and out of the nucleus. But he had one good copy, one bad copy. The therapeutic hypothesis is pretty straightforward. Let's knock down the bad copy and hope that the good copies provide sufficient transport in and out of the nucleus to be helpful. The patient was presenting with seizures and sort of having real trouble. They found us about 7 months after their diagnosis, this is never a fortunate encounter in that sense, but it was a counter that provided from synergies, which is to say we were in a place where we could say, yes, we'd like to try to help. So we tried to help, and in about 5 months after we fired up the platform, and this is a place where I think collaboration needs to be smoothed out. We waited for a month, maybe a little longer, to get some cells out of some academics, to make sure we could do the experiments that we needed to work with. And then we were able to push forward and start doing the experiments. We ran in parallel, of course, the activity study and the safety pharmacology studies. And we were able to run it with a very small number of compounds because we had high prior that these compounds should be safe based on our models. At the end of the day, the compounds work pretty well. What I show here on the left graph is the relative expression measured through an NGS assay. And you can see we have a control oligo, doesn't target anything really, and it's pretty flat line. It tells you two stories though. One, an oligo doesn't do anything to impart the sort of allele expression of this gene if it's not designed to that particular oligo. And the patient fibroblasts here express the gene at about 55%, the reference copy or good copy of this gene. When we dosed with our lead compound, we're able to drive that up to about 90 to 95% good copy, 10%, 5% bad copy. That's exactly what we want to do. It's doing what it's supposed to do at a molecular level. But of course, we had to make sure we didn't get rid of the good copy. This just tells you the relative expression. So we also normalize expression. This is measured with qPCR, then overlaid with the NGS data to sort of impart the relative expression of each allele, where the pathogenic allele is colored in red and the reference or good copies are colored in green. Apologies for the colorblind. That's the bad copies on the bottom, good copies on the top. And what you can see is that as we increase dose, we get a very nice knockdown of the gene to about 50% control. We even see the cell doing what we hope it do, which is it realizes it doesn't have to jock copies and starts to upregulate. But what's really telling is, of course, that we knock down mostly the pathogenic allele. That loss of expression is almost entirely accounted for by loss of pathogenic allele. And then, of course, we are thrilled that it cleared through all of our tox studies. One thing I'll say is that when we do these tox studies, we often think about the tox studies as the mice are really sort of, and we think about, we inject them into mice. This is going to be an intrathecal administered drug, but we're worried about systemic toxicities. Why? We worry about systemic toxicities because we think that the organ systems, when you expose them to an oligo, are kind of like canaries in the minefield. It's hard to see things go wrong in the brain because the brain's complicated. It's got a lot of things going on. You don't have a lot of bioavailable markers. But if you put it systemically and you see ALTs go up, you know you're hurting hepatocytes. Will it hurt neurons? Maybe not. But if it hurts hepatocytes, it's probably not the best drug. I don't want it in my brain. So we think we use every one of these sort of biomarkers as a systematic way of being sort of canaries to see where there may be warts in an in vivo setting for a drug that could be going into a patient to ensure that we get as much safety as we can on each one of these compounds. And the sort of happy ending at the end of this is that in about 5 months we went from whiteboard to compounds where we were fighting about which compound to advance forward because they all look pretty good. We then scaled up for GMP and GLP studies. This is required, of course, to get something into a patient. We then filed an IND after we got the data back, managed to get that IND approved to dose, and then we were able to dose the patient. I'll tell you, in this process, we had hoped that we'd be doing this in a way that would allow us to have a little extensibility beyond an N of 1, if you will, and we'd hoped to be able to sponsor this, but the patients, the interactions and the patients' symptoms were getting worse. And the regulatory path was not emerging fast enough to do something different. So we fell forward and we used the N of 1 guidance that allowed us to get this into a patient as fast as possible. And I'll say, and we'll have more to share on this, I'm sure, in the future, but insofar that patient's been administered for about 6 months and that drug has been exceptionally well tolerated. We have no safety signals, and we are seeing significant benefit as we look at the various different markers of that clinical trial. And I think that highlights where we're aiming, which is, and I think we all see this, right? We see exactly Francis Collins' notion, which is we can understand the molecular pathology of disease now. And we for rare diseases, it's often super clear because it's monogenic and we can drop them down. It comes out of a single sequencing event. For common diseases, this is coming forward as well. We get to a place very rapidly now where we have actionable understanding of this disease biology. What we want to do is we want to connect that dot with a platform that allows us to engineer these compounds in a way that we can get treatments to patients in a way that's efficient enough to make the sort of commercial model about whether you're treating 1 patient, 1,000 patients, 100 patients, or 1 million patients, sort of an irrelevant conversation. It should be about what's possible. If it's possible and you know you can have benefit, we should be able to make a drug. And then it has benefit, payers should be able to reimburse and we should move forward. And I'm looking forward, and I know this session was a little bit about how do we bring an ecosystem together. And I think part of this is making sure that everyone in this space is able to work together and find places where we can have synergies and be helpful in terms of making sure that as we find paths, we can build off on each other's successes. So with that, I'll sign off.
Thank you, Grace.